This is the presentation of Ashton Munoz and Alison and our data contribution to an educational study of applying bioinformatics modules to undergraduate learning. Our rodent and bat research were conducted in the INBRE Mole Tech lab at the Northwest College in Powell, Wyoming over the span of 2 semesters. Hello, my name is Ashton Munoz. I'm an undergraduate nursing student co-enrolled at Northwest College of Powell and the University of Wyoming. I'm married to Spencer and we have 2 children and 2 dogs. I will graduate with my Bachelor of Science in Nursing in the summer of 2023 and plan to earn my Master's of Nursing while residing in Colorado.
Hello, my name is Alison. I am an undergraduate nursing student co-enrolled at Northwest College of Powell and University of Wyoming. I have 2 children and 2 dogs. I will be graduating with my Bachelor of Science in Nursing in summer 2023. From there, I will earn my Master's in Nursing and pursue a career as a pediatric nurse practitioner.
Our core focus while students of INBRE was to test and analyze fecal samples for pathogens with the potential to infect human hosts. We looked at a variety of small mammal samples including house mice, deer mice, white-tailed prairie dogs, bushy-tailed wood rats, cottontail rabbits, and little brown bats. These samples were collected from Wyoming, Montana, and Minnesota. We were mostly interested in studying Yersinia pestis, but were unable to achieve this task as that would have required a level 3 containment lab. Yersinia pestis has the potential to be weaponized and our lab couldn't accommodate its presence. While we didn't find Yersinia pestis in our samples, we did find a broad range of other human pathogens to study. When we first began working in the lab, we tried to see the DNA from our samples using a procedure called electrophoresis, which works by applying a negative charge to a liquid medium, causing proteins in the DNA to move towards a positive charge. When done correctly, the blue DNA moves in a horizontal path across the gel medium. Our experiment was unsuccessful. While working in the lab, we used a Powell fecal DNA extraction kit, which consisted of 7 liquid solutions used to wash the DNA out of the sample, a centrifuge, and a qubit 4 fluorometer, which measured the concentration of DNA in our sterile extracted samples. We froze our DNA in this extracted state for later analysis. We worked in the lab 1 day a week with our mentor, Dr. Alison, a microbiologist and retired assistant professor of biology at Northwest College. She directed us through the careful process of extracting microbial DNA from stool samples and removing any potential bacteria or fungi in the preparation for further downstream testing. We measured out a small fragment from each of our extracted DNA samples for genetic sequencing and sent them to the Hubbard Center for Genome Studies at the University of New Hampshire. This allowed us to see what types of DNA were in our original fecal sample, including organic material, in particular vegetation from the mammal's diet, as well as zoonotic pathogens.
After our samples were sent to the University of New Hampshire, we were given 2 files to decode. Prior to deciphering the codes through the RON system, I was able to open them to see what the DNA sample results look like. This is not even 0.5% of the DNA sequences for one sample of a basic house mouse, Mus musculus. Once the metabar coding was done through RON, the files were able to be downloaded to the computer, which we then analyzed through the CHIME website. These are the demuxed reads and Fred quality scores of the samples. In order to more accurately organize and decipher the data, we then had to trim the sequence base of the reads. After trimming data, this shows several possible identifiable bacteria as well as a few that were unidentified. On the bottom of the chart, the samples are listed by species: prairie dog, cottontail rabbit, wood rat, bat, and 2 types of mice. For these results, we pulled the 16S ribosomal RNA data. The demuxed raw reads for the bat data shows good and bad Fred quality scores. The first trimming attempt was unsuccessful and yielded no results. The second trimming attempt yielded better results with a different graph. There were multiple bat raw reads that were too unstable for analysis, as seen in the dark region of the data plot. There are many unidentified bacteria types. 2 house mouse samples were analyzed incorrectly and present in the data with bats. The bat data for 16S ribosomal RNA is varied from the bat data obtained using the 18S ribosomal RNA. This is also true of the house mouse.
We would like to thank Eric Atkinson, Elise Kimball, Gary Wallace, and Jay Dickerson. We owe you our knowledge and credit you with our passion for DNA. We would also like to thank the INBRE grant contributors for funding our research program at Northwest College.